Hi everyone again. Welcome to MBG Turkey Agenastro 4th International Astrobiyoloji Conference. This is today's third and last session. I'm Zeynep. You know me from the past years, but I want to introduce myself again. I'm the head of Agenastro team, and I'm a master's degree student in microbiology department of Medipol University. And today we will welcome Dr. Bruno Cuevas Zuviria in this session. And he was a postdoctoral researcher in Betül Kaçar's laboratory of Wisconsin Madison University. He has recently returned to Biocomputation Laboratory. Today, he will present us their recent article about emergence of an orphan nitrogenase protein following the atmospheric oxygenation. So, hi, Bruno. Welcome. How
Hello.
are you?
I'm fine. I'm excited. Thank you for the kind introduction.
Thank you. Do you want to add anything for your background?
No, I just have to say this is the second time that I'm invited to give an online seminar
Yeah.
for Turkish audience. So I really have the hope that for the third time I'm invited on site, so I can really I can visit Turkey for the first time. But yeah, until then I'll keep. It really makes me very happy to be here today.
Oh, thank you. It's nice to welcome you here. Also, I'm really thankful that you accept our invitations. So, if you are ready, you can start for your presentation. Stage is yours.
Great. Hi, everyone. Well, as I say, I'm very glad to be here today. Today, I'll be talking about one of the projects that I did during my time in the lab of Professor Kachar in the University of Wisconsin-Madison. I guess, like most of you might know who Professor Kachar is, but for those who don't, Professor Kachar is a pioneer researcher in the field of astrobiology and the origins of life. So before starting, a couple disclaimers. First, I recently moved from the lab to a different position, so I'm not really right now a core member. So I only represent myself here and the work that I did there. And second, I'm gonna give a quite general talk about the nitrogenases and the challenges, the results of the lab. This doesn't mean that I did all the work that I'm presenting here, because this is a very multidisciplinary, very broad work that is done by many different people, and I play a part there. I do mostly the structural computational analysis, but there are other parts that are done by great researchers that do experiments, that do microbiology, etc. And those I'll try to point their contributions as much as I can. So before starting, I want all of you to assume that we want to know how Earth looked like at the beginning and how life emerged, evolved, and thrive in the early planet. Of course, this, given the spirit of the conference, I guess like you already do. Well, in my case, I wonder about how life evolved and thrive. But when I say about how it did in the past, I'm not talking about like dinosaurs or mammoths. I'm talking only about this part of the history, which is the Precambrian. Oh, I guess like you cannot see my pointer, but anyway, I'm talking mostly about the Precambrian, which is a very long. A very long chunk of time between the formation of Earth and the emergence of the first fossils. And during this time, we have a very big challenge to understand life. It's like there are barely any fossils. Like most life was microbial back then. So it means like it didn't leave a very big fossils that we can analyze in the lab, or that we can observe, that we can extract. So it means like we don't have that resource that help us understanding life in the past as we do with dinosaurs or trilobites or whatever. So that means that we have to be a little bit more creative. Luckily for us, there is like a couple ways to look at this. One is to look at the geological record. We're lucky that some rocks, especially sediments, have some traces of the living organisms that were living on them. So sometimes looking at rocks that are very old might help us understand a little bit our planet in the past. And second, biology. All of organisms are vertically related, so that means like there is a little bit of the first organisms in all of us. And so this is an information that we can try to use to understand the first organisms. So in our lab, we try to understand our planet better through the resurrection of ancestral proteins, something that is possible thanks to a technique that is called ancestral sequence reconstruction. And we try to combine that technique with computational analysis and synthetic biology. I like to make a stop here. When I talk about the ancestral sequence reconstruction, I would like everybody to have a very clear picture of what this means. So nowadays, for instance, there is like a lot of sequences that we can obtain from, let's say, from organisms that have their genome sequence, from metagenomics. And those sequences can be aligned to generate an evolutionary model of the given protein, for instance. And from that, we can obtain a phylogenetic tree. A phylogenetic tree is telling us somehow like the story of those sequences, like how they evolve from each other, how they diverge, etc. And in these phylogenetic trees, there are some nodes here that represent the ancestors. Those ancestors are extinct. They don't exist. They are not with us anymore. But thanks to some statistical techniques, we are able to reconstruct the sequences of those ancestors. And once we have those sequences, we can, as I said, we can use synthetic biology tools to try to resurrect them, put them in the lab, etc. So today, I'm gonna talk a bit about elements. As you see, this is the periodic table. We have 100 elements, a bit more than 100 elements. Some of those are relevant for life. Some of those are not very relevant for life. And today I'd like to focus especially in one element, which is nitrogen. Nitrogen is in almost all biomolecules, and it's fundamental for life. It's part of the famous CHOMPS acronym. However, it's also kind of problematic. Nitrogen on the atmosphere is quite abundant, but it's only in the form of N2. N2 is very unreactive. It has a triple bond that makes very difficult to make any reaction with those nitrogen atoms. And right now, life has only found one way to turn that nitrogen from the atmosphere into nitrogen that can be assimilated by living organisms, which is these arrows here that we see called N fixation, carried out in this case in this figure by Neef. I also have to say, and it's important for us to understand that there is not really a very big abiotic counterpart for nitrogen fixation. So that means if we remove living organisms, there are not really that many processes that can fix nitrogen, or that can do it on a biosphere scale. So this reaction is carried out by the nitrogenase, which is a quite complex and remarkable metalloenzyme. It is carrying out this reaction that you see here in the top. As you see, a nitrogen molecule becomes 2 molecules of ammonia after adding 16 molecules of ATP and 8 electrons. So as you see, this is quite expensive reaction. It's carried out by this enzymatic complex that you see here. It has 2 components. The component 1, which has the catalytic component, and then 2 subunits. The component 2 that is a component that only provides energy and electrons for this reaction to continue. If we focus on the component 1, we can see that there are 2 metal clusters in this protein. We have the P cluster and the M cluster. Well, the M cluster is maybe one of the most remarkable metal cofactors ever. It's this kind of weird origami that you can see here. Has like a central carbon, a lot of iron atoms, which are like the orange atoms, and sulfur atoms. Is coordinated with some residues in the nitrogenase, and is also bound to this homocitrate molecule, which is also part of the cofactor. If you realize here, there is an atom here that is painted in teal or in gray. That atom is quite important because depending on the element that we find in that atom, we have a different kind of nitrogenase. There are nitrogenases that require a molybdenum atom in that position, and those are the molybdenum-dependent nitrogenases, or NIF. Actually, they are the most abundant, the most well-characterized, and the most productive ones. And then we have the vanadium and iron-only nitrogenases, which are a little bit weird. We don't know so much about them, and they are like way less characterized. The chemical reaction that this nitrogenase carries out is pretty complex. As you saw in the previous slide, it requires to place 8 electrons and to break 16 ATPs. Of course, that's not made in a single step. It's actually made in many rounds of reduction. Each of these rounds of reduction can actually go back. So like this reaction is partially reversible. And as a result of that reversibility, we have that nitrogen fixation is usually leaking hydrogen. So the reaction that I gave you here is actually an ideal reaction, but this always happens. This is never as efficient as I'm showing you here. And actually, the alternative nitrogenases seem to be way less efficient than the molybdenum nitrogenase. There is also the need to bring many electrons and to hydrolyze a lot of ATP, and that's done through the binding and unbinding of the component 2 and the component 1 in something that we call the iron cycle, which is the way to provide these electrons and energy. Then we also have that this enzymatic complex has all these many metal cofactors that, as I showed you, they are quite complex. So here, for instance, I'm showing you a figure of a paper that describes all the proteins that are associated with the NIF nitrogenase in Azotobacter vinelandi, which is the main model organism in nitrogen fixation. And as you can see here, HDK are the subunits of the nitrogenase, and all the others are proteins that are required for this protein to work, but are actually not part of the component. So this kind of tell us like how complex is the biosynthetic pathway that we need to be able to generate those metal cofactors. Here on the right, I'm showing you, for instance, a diagram of how to generate the metal cofactors. And as you can see, it's actually quite complex. Oops. Finally, I like to remark that nitrogenases are actually quite, they have a bad relationship with oxygen. They are metalloenzymes, and the metal cofactors that we have in there can be deactivated by oxygen. And that's quite a problem nowadays, given that oxygen is a little bit everywhere. So this means like the organisms have to adapt to the presence of oxygen through many different mechanisms. For instance, here on the left, I'm showing you a figure that shows how the organisms that have adapted to make nitrogen fixation in the presence of oxygen have more proteins than those that do nitrogen fixation in the absence of oxygen. Some of those proteins might be oxygen protection proteins. Then we also have that some organisms have specialized activities to protect from the nitrogenase from oxygen. For instance, in the case of the Azotobacter vinelandi, we have respiratory protection, which means like the cell is just burning oxygen as fast as it can, so it can protect the nitrogenase from oxygen. We also have that some prokaryotes actually have some sort of cell specialization. In the case of filamentous cyanobacteria, and they have these special cells we call pterocysts that don't do photosynthesis, but they do nitrogen fixation. So that way they separate these two activities that will be harmful to each other. And finally, we have symbiosis. Symbiosis is quite abundant. For instance, we know that the Rhizobia bacteria make a symbiosis with plants, and they live in the roots of plants and provide nitrogen for them. And now this week, there is this publication in Science that claims that that they discover a guy that has a bacteria inside that works as an organelle and is fixing nitrogen. So if nobody finds any anything against this. This means like this could be maybe the discovery of the year, maybe because this is the first time we find eukaryotes doing a nitrogen fixation. I mean, it's pretty recent, but I'm pretty. I think that we are all like quite fascinated by this discovery. So now I told you that nitrogenases are quite. They have a problematic relationship with oxygen, and this allow us to go back to the past. Like let's try to see what does it mean. What does this mean at the in the level of the history of the Earth. So ancient Earth is like the Earth we find like at the beginning of the Archean is actually assumed to be anoxic. So there wasn't barely any oxygen on the surface. And the geological and biological record indicate us that there was probably a major event around 2.5 giga years ago in which molecular oxygen appear on the surface of Earth. And when I say appear, it doesn't mean like it gets to the current levels. That actually it only did that a few million years ago. But at this time, it was only an increase to a 1% of today's levels. However, that transition from not having any oxygen to having a little bit of oxygen is actually quite dramatic. And it could have had a very big effect on the Earth's surface. Among other effects, for instance, we have the reduction of methane levels, which could have frozen the Earth, given that methane was an important greenhouse gas. Then we also have the emergence of oxidation, like living organisms have never met oxidation before, or like reactive oxygen species. So that means like they had to adapt pretty fast to this new harm. Also, we have that many enzymatic activities, as for instance, the one that we are talking about today, became affected by the emergence of oxygen that will inactivate their active sites, etc. And the emergence of oxygen, it will also affect the concentration of certain nutrients that are required for life. For instance, like we have that the emergence of oxygen will have meant that a lot of sulfates will have become sulfates that are soluble. So that will mean like there will be a general increase of the level of sulfur in the oceans. But it will also that will also mean that there is a change on the concentration of different metals depending on the relationship with sulfur and oxygen. So, for instance, iron, which was very abundant back then, but it precipitated during this great oxidation event, forming these iron band formations that we can find in many places. And nowadays, it's a very scarce resource, a very scarce metal in our oceans. On the contrary, molybdenum, that metal that atom that makes that we find in the most important nitrogenase, it actually went from being very scarce to quite abundant if we compare it with all the other metals. So this leads me to the first question that I would like to debate today. What is the? I mean, as I told you before, molybdenum nitrogenases are the most abundant ones, but we also have some nitrogenases that don't require molybdenum. But we also find that there is molybdenum was pretty scarce in the deep past. So will this mean like the first nitrogenases were those alternative nitrogenases that we know very little about? Well, the answer to that question comes from a couple sides. A couple avenues. Sorry. The first one is the avenue carried out in our group, the phylogenetic analysis. Here, in our group, a phylogenetic tree was built using the nitrogenase subunits, and it indicated that iron and vanadium enzymes, nitrogenases, come from a molybdenum-dependent group that is actually not very well characterized. We don't know a lot about what that group is doing. Then in this same study, there was a machine learning based prediction applied to the ancestral sequences, those sequences that we can infer from our phylogenetic tree. And what it actually provided is that all the ancestors of the nitrogenases, once we get out of the last common ancestor of the alternative nitrogenases, use molybdenum. So this means like life, even if molybdenum was very scarce at the beginning, still tried to use molybdenum to make a nitrogen fixation. This result that comes from our computational analysis is also confirmed by looking at the geological record. Here, they analyze the isotopic signatures of nitrogen in sediment rocks, and they found that there is like some of those sediments have isotopic signatures that match better the molybdenum-dependent nitrogenase than the alternative nitrogenases. Besides, those actually correspond to measurements of rocks that are 3 to billion years old, which means like we have nitrogen fixation quite early on the Earth history. For those who don't know what is an isotopic signature, let's say that enzymes have a certain tendency to choose some isotopes over others. So this is a little bit what we are measuring here. So these two lines of work kind of prove that the first nitrogenases were molybdenum-dependent ones, even though there was not a lot of molybdenum. So right now I show you like a computational analysis, and that's cool. But I guess like you all might agree that it will be cooler if we could actually not just doing a computational analysis, but really put those enzymes in the lab and resurrect them and see what they do, right? So there is this pioneer work and this done in the Kachar lab to resurrect a nitrogenase, an ancestral nitrogenase. So for that, first there was a phylogenetic tree inference followed by the cloning of the ancestral sequences that we obtained from that tree. Then the cloning and the expression of those clones in the Azotobacter vinelandi, which is, as I said, the model organism for nitrogen fixation. Those were, and then that Azotobacter vinelandi with this ancestral protein was characterized, and we also carried out some purification and biochemical analysis of the ancestral nitrogenases. As you see here on the panel C, one thing we did was to prove to try two different ancestors into different approaches. The first one is just to replace one of the subunits of the nitrogenase, for which we use very recent ancestor or a little bit older ancestor. And in the second approach, we just try to use like all the subunits of the ancestor. So this is a work that was carried out before I arrived to the lab, but or during while I was arriving to the lab, but didn't contribute to this one specifically. But anyway, this work was actually successful because it proved that these ancestral nitrogenases were able to grow in this extant Azotobacter vinelandi, and it also show us that there is actually that this work is possible, right? So that those ancestral nitrogenases can be expressed, and they can actually have similar properties in some cases to the ones that we observed today. Now I'd like to come back to this to the phylogenetic tree, because as I said, there is like a lot of questions in this phylogenetic tree. Here we can see the alternative nitrogenases. They are like a clay. They emerge as a clade of a molybdenum nitrogenase, and we are still interested on them because they are, how we say, they are pretty weird. I mean, I still don't understand them very well. First of all, we know from our phylogenetic studies and from the isotope analysis, etc., that they evolved later. So even if they are using some metals that were more abundant or so, they came later. They also, in some cases, for instance, in the case of the iron nitrogenase, iron-dependent nitrogenase, it uses a slightly simpler pathway to assemble the cofactor, but it still came later. Then they are never present in the absence of molybdenum-dependent nitrogenases, so they are always working as some sort of backup enzyme, as I'm showing you, for instance, in this study on the left, where we can see that there is always molybdenum nitrogenase, even in all the organisms that they analyze. They are less efficient. They tend to leak more hydrogen. They are also able to reduce other substrates. For instance, like they are able to reduce methane in the case of the iron-only nitrogenase. And we don't really know what is the physiological significance of those reductions, whether they are like more like a lab artifact or they have really like a function. And then they also seem to have a different stoichiometry with an additional subunit that I will talk about in the next slide. So in general, like alternative nitrogenases have a lot of weird things that we don't fully understand. And I think it's also important to remark their significance in astrobiological terms. We're talking about enzymes that use different elements. So let's say if we visit the planet and we find there is no molybdenum, should we discard the search for life? Or, I mean, could have life found a way to fix nitrogen in using these different enzymes? So today I'll focus, now I'll start talking about the article that we just published. It's been a long path for publication, but in this study we were working on the G subunit, which is this strange subunit that we find only in alternative nitrogenases. So here in the panel A, I'm showing you the two different nitrogenases, the alternative nitrogenase BNF and the molybdenum-dependent nitrogenase NIF. So as you can see, there is this subunit, the G subunit painted in dark blue, that is only found in the alternative nitrogenase. It's bound to the D subunit. It doesn't have a metal cofactor. It's not really interacting at the active site. It's kind of small, around 100, from 110 to 130 residues. And it has this structure composed of 4 alpha helixes that was not discovered until quite recently, 2017. And as you see in panel C, is also expressed in the alternative nitrogenase operon. So we don't really understand what is the role of this G subunit, but we find it in all alternative nitrogenases. So does this mean that it enables these nitrogenases to work with a different metal? And if it does so, how? Well, this is a pretty controverted, this is still like something we are thinking about. Indeed, there were like already some ideas about what could be the role of this G subunit. For instance, there is this previous study where they considered that it's possible that the G subunit could be mediating on the recognition of protein-protein interactions between the component 2 and the component 1 of different isozymes. So in this case, the G subunit will be just like a gatekeeper saying, hey, I interact with you or not with you. However, this is not super clear, because in the end, like, these proteins also have their own cycle, the iron cycle that I showed you before, where the proteins tilt, as I'm showing you here in this figure from a recent article. So it's not fully clear, at least for me, that this might be the role. So we decided to make our own questions here and try to answer them. So today I'll go over those 5 questions and I'll show you the results that we obtained. Oops. So the first thing that we wanted to understand is, okay, if we have a G subunit that we don't know what it's doing, can we just look at its relatives at different proteins that are related with this G subunit, see what they do, and try to infer what the G subunit does? So this idea of like guilty by association will actually help us quite a bit, right? However, we were pretty unlucky here, because it seems like the G subunit doesn't have any relative. To try to assess this, we tried different methods. We used BLASTP to detect the homolog proteins. This kind of failed. We didn't recover anything that was not a G subunit. We also tried T-BLAST-N to try to find homologs of this protein in non-coding regions. So in case like protein annotation, genome protein annotation failed in the case of the relative of the G subunit, but we didn't find anything. We also use ZBLAST and Hammer to find very far homologs. So in the case that the G subunit homolog kept evolving for a lot of time. So right now they are very different, but they are still doing the same or something like that. But we didn't find anything. And additionally, we also tried a structure-based method to find homologs. This is actually pretty new. It's based on the idea of mining the AlphaFold databases that have appeared a few years ago. But in this case, we also failed to find any kind of structural homology. So this means like, after trying all these methods, we didn't find any meaningful hits. So we concluded that this G subunit is an orphan protein, so it could have no homologs, at least that we know. Of course, it's always much harder to test that something doesn't have homologs than to test that something has. But given this result, we wonder like, how is this possible? Like, after all organisms are vertically related, right? So we should be able to find a relative for the G subunit. And there's actually a process that is called de novo emergence that is actually kind of characterized in eukaryotes that have very large genomes with a lot of non-coding regions. So in those cases, it's been possible to trace the origin of some orphan proteins to de novo genes that start expressing in non-coding regions. But in the case of prokaryotes, this is actually extremely challenging because, after all, prokaryotes have very streamlined genomes, so it's very hard to find very large non-coding regions. And if there are those non-coding regions, it's very likely that they are lost pretty fast. So then, if we cannot really assess the function of the G subunit by looking at its relatives, we will have to just look at the G subunit and see what we can find. Luckily, we can try to look at the past, and that's actually what we do the best. So we started by trying to inferring the phylogenetic history of the G subunit. So here I'm showing you a phylogenetic tree of the alternative nitrogenases rooted on the molybdenum nitrogenases NIF1 and NIF2, which are the main nitrogenases that we find in most organisms and most nitrogen-fixing organisms. So as you see here in this phylogenetic tree, we also mapped the optimal growth temperature of the microorganisms having these proteins and the tolerance to oxygen of those organisms. So as you see here, there is this group of what we call NIF3 that is the clade that from which the alternative nitrogenases emerge. This clade is actually mostly composed of organisms that are sort of extremophile, that grow at very high temperatures, and they are mostly anaerobic. So we could be talking here about, for instance, hydrothermal vent organisms, methanogens, etc. And then we have the alternative nitrogenases organisms that, as you see here, they are mostly mesophilic, and they can be both aerobes or anaerobes or facultative anaerobes. So that means like they have a much wider niche location. And here we can see all kind of organisms. So there is not really like, there is like cyanobacteria, alpha proteobacteria, delta proteobacteria, chlorophylexes, etc. And one thing that we find when we were building this tree is that actually none of the organisms that have an alternative nitrogenase have a NIF3 alternative nitrogenase, a NIF3 molybdenum nitrogenase. So as I said before, all alternative nitrogenases always show up in the presence of a molybdenum nitrogenase, but never in the presence of this group that we see here, the clade NIF3. So this could mean that somehow the alternative nitrogenases are the evolution of the NIF3 clade to be that can exist in a wider environmental niche than the original NIF3. We can also see that the G subunit is only found at the root of the alternative nitrogenase clade, which is something that we could expect, given that all alternative nitrogenases have a G subunit, and none of them molybdenum-dependent nitrogenases have a G subunit. And here we're highlighting some of the ancestors, the iron and iron-only dependent nitrogenase, the vanadium-dependent nitrogenase ancestor, the vanadium and iron ancestor, and of course the previous ancestor that in this case we call NIF-ANC, although it's not really the ancestor of all NIFs. Those are the ancestors that we can actually that might tell us something about the origin of this G subunit. So using our ancestral sequence reconstruction tools, we can generate their sequences. And nowadays, if we can reconstruct their sequences, that means we can also reconstruct their structures thanks to the latest deep learning-based structural prediction methods, as for instance, AlphaFold. So we use these structural prediction tools to try to understand what it was the molecular context in which this G subunit emerged. So after obtaining structural models, quite reliable structural models for these ancestors, we look at their structures, and we look especially at the regions where the G subunit will have emerged. So as you see here, we have on the left the ancestor previous to the emergence of the G subunit and the alternative nitrogenases. And on the right, we have the first last common ancestor of the alternative nitrogenases. So one thing that is called our attention is that when we look at that interface, there was a bunch of residues that were quite conserved on the surface of the D subunit. And it seems like those residues are actually involved on the interactions with this G subunit. This is actually not very common because most protein surfaces are not very well conserved. I mean, after all, they are exposed to solvents, so they don't really have any important interaction. But in this case, they were pretty conserved. So that tells us something about the role of that surface. It's very likely that that surface was already a hotspot for protein-protein interactions that we either don't know yet or that were lost in the past. So that means the G subunit will have bound to a place where other proteins might be binding. And which proteins will those be? Well, actually, when something that has been characterized in the NIF nitrogenases, in the molybdenum-dependent nitrogenase, is that the metal cofactor that, as I told you, has a very complex biosyntactic pathway, is inserted into the pocket of, to the active site where it should be by an enzyme that allows to open the D subunit, creating some kind of tunnel where this metal cofactor can be introduced. So that metal cofactor tunnel, insertion tunnel, is actually pretty close to the location of the G subunit, and it actually has some kind of entrance where we find that there is this residue, the leucine 18, that I'm showing you here with this arrow, that is actually conserved in all G subunits. And this is actually quite a remarkable feature because most residues in the G subunit are not very well conserved. So it seems like this leucine sits there in that kind of pocket that sits pretty close to the metal cofactor. So it's probably that whatever role the G subunit has, it could be related with the metal cofactor insertion, either trying to prevent the exit or the entrance of the metal cofactor, or doing something alike. Finally, as we said, there are many nitrogenase, many alternative nitrogenases ancestors here that we can study. And one of the questions that we had when we were doing these experiments is okay, how much of the G subunit is actually specific to each of these iron or vanadium isozymes? For instance, will they be swappable? So using the ancestral sequences, we synthesize the genes corresponding to the ancestor and also to the iron-only nitrogenase, and we try to put those on a vanadium-dependent Azotobacter vinylandi nitrogenase. So as you see here, what we did is first to knock out the G subunit, and then once we have knocked out that G subunit, we introduced it at plasmid that either had the ancestral nitrogenase or the iron-only nitrogenase or non-nitrogenase in case to provide a control. Our results actually show that there is a decrease on the nitrogenase activity And actually, in the case of the ancestor, it seems like we don't that there is not significant differences between our knockout control and this ancestor. So it might seem like it's not really functional right now. So this means like things have evolved, and we need to figure out why it's not working. But in the case of swapping the alternative nitrogenases, we find a reduction, but it's still like a functional nitrogenase, which might be due to the reduction on the protein-protein interactions that we see between these two enzymes. I mean, between these two proteins. So here I'm showing a recap of our conclusions in this study. So in the end, there is this NIF ancestor, like the previous ancestor before the emergence of alternative nitrogenases. This ancestor at some point bound to this ancestor of the G subunit, which emerged de novo, and it gave place to this ancestral last common ancestor of the alternative nitrogenase. And this one then specialized into two different alternative nitrogenases, which use vanadium and iron-only cofactors. Also, our study suggests that this event might have taken place during the Great Oxidation event. That, as I told you before, means that there was like a huge change on the Earth's atmosphere, pretty dramatic change, which might have this gave place to a lot of adaptation in many different enzymatic systems. And in this case, we think like providing an alternative nitrogenase could have allowed organisms to adapt to more different niches, where they could be like different concentrations of metals, etc. So with this is my seminar. I think I finished more or less. Well, I've been a little bit over time, but I'd like to thank everybody. I mean, as I say, I only did the structural computational part. This article that I'm publishing here is actually, I published it, is co-author with Amanda K. Garcia, which is the expert in phylogenetic inference. Then we also have some of the experimental lab members that did all the experimental work, and of course our PI, Professor Betukechar. So this is a pretty wonderful team that has taught me a lot about evolution over the deep past and science in general. I also like to thank my funding sources, because they allow me to keep doing what I do, which is science, and of course all of you for your attention. And just before finishing, I like to sponsor my seminar cycle. I mean, I also have my own seminar cycle, which is live. I mean, well, it's not mine, of course. I collaborate with a bunch of people to make it possible. So if you scan that QR code, you will find that we are doing seminars in English once a month, and we always try to bring speakers that work in different topics around not just the emergence of life, but also like how life started evolving from the first cells to multicellular eukaryotes. So if you like my seminar, I'm pretty sure you will like the seminars that we have there. Thank you everybody for your attention. I'll be happy to take some questions.
Okay, thank you for your this presentation. We learned a lot because it was really good research and also a good talk. And if it is okay for you, I can start with questions, because we have many, and if you have time, I want to cover all. First,
Sure.
I will remove the slides from here, and also we will share that QR code in our social media accounts, so if they want to watch your seminars, they can follow from there. Okay, so the first question, what are the main findings of the paper? How was the orphan nitrogenase protein associated with atmospheric oxygenation?
Well, for me, the two main things that we were discovering is that we were describing the evolution of this G subunit. I mean, so far there were some speculation about what this G subunit was doing in the past, but nobody had really tried to see how this G subunit looked like. We also show that there is a very interesting structural pattern. It seems like the D subunit was some kind of nest where we can plug the G subunit. And it seems like that nest was a hotspot for protein-protein interactions. Of course, finding those is a little bit more challenging, but we have some clues about which one it could be, considering, like, as I said, the story about the metal cofactor oxygenation. And then, of course, regarding atmospheric oxygenation, we think we have here a case of environmental adaptation. I mean, there is a lot of, just looking at our phylogenetic tree, we know that we can constrain more or less this event in the past, in the deep past. And there is some previous evidence by, for instance, the work from Rick Anderson that places the emergence of the alternative nitrogenases following the great oxidation. So in our case, we think like all the pieces of the puzzle kind of point to the possibility of this G subunit being an adaptation to this new environment that had a lot of hazards, that was very different from what biology had known before. And in the end, solving what is exactly the relationship with atmospheric oxygenation requires to fully understand what is the purpose of these alternative nitrogenases, and that's something we still have to figure out in general. But yeah, I think those will be like the main contributions of this paper.
Okay. So, in which organisms was the orphan nitrogenase protein found, and what are the evolutionary connections between these organisms?
Well, this orphan protein was found, after some rounds of experimentation. Once they had discovered that there were some alternative nitrogenases. Those alternative nitrogenases appear, I think, because there was like some contamination with wolframium. Wolframium is usually inhibiting the nitrogenase enzyme. It only inhibits the molybdenum nitrogenase, but they still found that there were like some strains that were able to grow, and they found, and I think that's how they found these alternative nitrogenases. Then there was some later research that found that there was this subunit that was required for this nitrogenase to grow. But of course, there was like a lot of there was been like a lot of debate in the past about whether that protein, for instance, like it was not clear whether that protein, like what was the stoichiometry of the whole complex, whether we have 4 G subunits bound to a complex, or if you look at some reviews from the past, you might find very interesting ideas about this. And we only get a crystallized structure of this G subunit in the 2017. Organism, we are most of the times when we are talking about nitrogenase, we're talking about Azotobacter vinelandi, because this is the kind of the workhorse of nitrogenase studies. The main reason is like this organism can grow in the lab without anoxia. It means it can grow on a bench, and that's a pretty big advantage if you're working with this organism, because that means like you don't need to get into these anaerobic chambers or anything like that. So I would say like 95% of what we know is about either Azotobacter vinelandi or some other organisms. But for instance, a few days ago, we were making an analysis. We were counting like how many structures have been placed on the PDB related to different organisms, and we found that there is actually only 5 organisms that have a component 1 crystallized. So that means like from all the organisms that do nitrogen fixation, we only know 5 structures, which is pretty interesting. And that's, in the end, most of the studies are pretty Azotobacter vinelandi centric.
Okay.
Just before, Azotobacter vinelandi is kind of a proteobacteria. It's not really it's not an organism that we can say, oh, it's very ancient. It's actually pretty recent.
Yeah. What inspired you to investigate the emergence of the orphan nitrogenase protein following atmospheric oxygenation?
Well, first of all, this is like a lab effort, right? I don't. It's not that I have these ideas and I do it. It's something we all start following. But in our case, yeah, we are very interested in how proteins relate with environment, and because, as I told you before, with the first story that I showed, like how we are able to know that the first enzymes were molybdenum nitrogenase. Oh, something happened with my screen. Well, I don't know. Oh, okay. Here it is again. The first organisms, the first enzymes that were, what I was trying to tell with that story is that there is a way to combine phylogenetic inference and how we say, studies coming from the biological side with studies that come from the geological side, right? And so I think like in the lab, we always try to think in both terms, right? Not just, and in the case of great atmosphere oxidation event, this one is quite a big geological event. It left a very big record, so it's usually a very interesting point of history. Just a side note, I guess you might have heard like the word the boring billion. The boring billion. That's
Yeah.
how it's called, like the geological time that came after the Great Oxidation Event, and that's
Yeah.
because not a lot of things happen that we know. So as you can see, like most of studies, in the end, we are always looking at this Great Oxidation Event with admiration. Like,
Yeah. Also, Betul Kachar has another article about sulfurase, something like that. I don't remember the name of article exactly, but she was also working on sulfur, so sulfur producing bacteria or something else. So nitrogenase is also not a surprise thing to think. I mean, as a laboratory.
Yeah. I mean, I cannot talk about all the projects in the lab.
Yeah, I know.
Like the Great Oxidation Event, it's something that we take a lot of attention from many different angles, not just. But that's all I can say.
Yeah. Okay, so how do you think emergence of this orphan nitrogenase protein impact our understanding of early Earth's nitrogen cycle? Actually, you have already mentioned this, but
for me, there's this another story that I'm trying to follow after this study. It's not so much about the nitrogenase, because, well, this one is a very specific example. It's very important. Without nitrogen, the earth will not be fertilized, and so on. But there is also another thing that I find with this story very remarkable. Is like we have a small protein that is binding to a larger protein and making it to do something different. In this case, using a different metal cofactor, but it's also making it do, for instance, reduction of different molecules. So I don't know. I think like this is like somehow an explorer pathway for protein evolution. That's something we also talk a bit, because this is not somehow like what we are usually told, right? Like, oh, we have this idea of like proteins just evolve one mutation at a time until they do something interesting or so on. And this is very like, okay, imagine like you had a Lego piece that you put on a Lego, and then Lego is completely different, right? So I think it not only gives me that not only give us that that new knowledge about the nitrogen cycle and the adaptation on the ancient Earth, but I think it also points to maybe some evolution knowledge, right? So
yeah. Okay. So this is from me. What are your plans for further exploration or questions that aim to investigate following this discovery? After that unit.
Well, I cannot say, because, as I say, I'm not a
I know.
Honestly, from the only thing I can say is like there's like many different interesting avenues here. I mean, the use of unit is something that gave us a lot of questions. Of course, there are other questions out there. So that's all I can say right now. But as I say, I think like there is also this evolutionary angle that maybe that's also an interesting thing for the future, not just like in the nitrogenase world. But so far, I cannot disclose much about what we do. I'm sorry.
Okay. Yeah, it's okay, because those further questions will be your next research. So I totally understand. How did the orphan nitrogenase protein emerge and evolve in response to the presence of oxygen in the atmosphere?
Well, I think like this is in line with previous questions, right? I mean, it's like we have this. Our idea is, we have this protein interface that was a hotspot for something else binding there. The use of unit bound there, and of course, we don't know all the pieces about how that happened. But it seems like that allowed this nitrogenase, which was a Ni3 molybdenum-dependent nitrogenase, to become something else, and that something else has been widespread distributed in a lot of organisms that exists nowadays. An important note here is also like it seems like alternative nitrogenases, despite being quite weird, they are still relevant for nitrogen fixation in many environments. For instance, the vanadium nitrogenases are actually prevalent in boreal regions where we have very low temperatures. So it might seem very low temperatures and the scarcity of molybdenum. So as I say, I think like these alternative nitrogenases might not just be like might be like an adaptation that allows organisms to be able to be in more niches than they were originally, and that might relate with the oxygen emergence, because oxygen emergence changed a little bit everything on Earth. So yeah, regarding the metal abundances, that's one of the things that we still like look at it. It's like, oh, this, you will think like the opposite. But if we look at the previous results that we have, we also found that the molybdenum nitrogenase was the first one, even though molybdenum was pretty scarce. So it's quite interesting. Like, it seems like these elements have really something on their own. They're not just chosen by nature because they're abundant. I mean, that's my take. But this is something that actually the lab is working on. There is this consortia that is actually funded by NASA called Muse. You Google it, you might find something about it. This is a consortia that is actually diving into these questions, like trying to understand what do they have, what do these elements have on the. It's like, are they really like, can life just take anything and make a nitrogenase out of it? Does it care about the abundance of these elements on the environment? These are the kind of questions that this group is trying to answer.
Yeah, thank you. Okay, next question. What ecological or physiological adaptations might the orphan nitrogenase protein represent in relation to the increase in atmospheric oxygen levels?
Well, I mean, so far we don't know what is the role of the orphan protein, right? I like to say there is not orphan nitrogenase. I mean, there is an orphan protein in the nitrogenase, right? Like the nitrogenase, we know where it's coming from. There is only a tiny piece that we don't know where it comes from, and we don't fully understand its role. We know it's there. We know it's necessary. So if you make a knockout, if the protein doesn't work, there are like some.
We don't know also the adaptations.
Yeah, and I can tell you about maybe the question was more related with alternative nitrogenases, and in that case, I can tell, as I said before, alternative nitrogenases, in some cases, for instance, we found that they are better for boreal environments. There is also some records of of alternative nitrogenases doing something. I mean, working on different environments. But so far, yeah, there are some gaps of knowledge there that we don't fully. There is also this quite interesting theory, this paper that was released a few months ago, about like the usage of about these alternative nitrogenases being a way to connect different organisms by providing energy. I mean, by liberating hydrogen. So maybe the hydrogen that we are seeing as a side product is not really just a side product, but it's a way for organisms to get rid of reducing power, getting rid of electrons or something like that. But that's something kind of hypothetical, but maybe that's a possibility.
Okay. So next one, what are the long-term effects of environmental factors such as atmospheric oxygenation and metal availability on the nitrogenase enzyme family?
Yeah, well, I mean, I think we can put this like more general, right? Atmospheric oxygenation, for instance, make iron scarce. If you look at every organism right now on Earth, all organisms are fighting for iron, right? Iron is like the limiting factor. Actually, if you look at the immune response, there are like a lot of mechanisms that in our body to try to deprive pathogens from iron, right? So this is like a fight that we've been since the Great Oxidation Event, probably. Like, it's very likely that organisms back then didn't have to worry about iron, but now we do. So that would be a a case of an example. I mean, of course, even though I talk about molybdenum and vanadium and iron, all nitrogenases depend on iron. I mean, actually, most of the metal cofactors are made out of iron. So that will be obviously impose a constraint. Then metal availability. We know, for instance, like terrestrial environments, they are deprived in molybdenum. So maybe that's a reason why that's one of the reasons why we find these alternative nitrogenases in terrestrial environments. But it doesn't make complete sense because we still find the molybdenum nitrogenases there. There is no organism that has got rid of the molybdenum nitrogenase, right? So that's it wouldn't. I mean, it will only make sense if we were able to find like a vanadium only fixing nitrogen organism, but that doesn't happen in nature. So we are not 100% sure yet about what is the role of these enzymes. So as I said before, there are like different factors that affect the evolution of this enzyme family, and of course metals is one of the most important ones, but it seems like there is some corners that we still need to to understand here.
Mm-hmm. I hope in next years maybe you can discover more.
Yeah, sure.
What significant contributions does this study's findings make to molecular evolution or biochemical research?
Yeah, I mean, well, I think I already indicated a few.
Yeah.
I mean, I already answered the question.
Yeah. That was the last question, actually. So I just wanted to show. So I'm really thankful for your time and your talk. It was really, really, really good. And I also want to thank you to accepting our invitation and coming here. So I am really appreciated when you accept our invitation. Because
Yeah, for me it's an honor, and I'm waiting till I get like on site invitation, so I'll keep accepting until they take me to Turkey. Maybe after that, maybe I will. No, just kidding.
Yeah, you can
Yeah.
come. And also for our next events, we want to see you again, maybe with your new research or new articles, or maybe if you want to talk about another things, we want to welcome you again.
Yeah, actually, I think I mentioned at the beginning. This is a work that I made in collaboration with a group, but I do other things too. So it's not my only kind of research.
Yeah, I know.
As I say, in this G subunit, I think this is a story that I really like, and this is actually a pretty recent result. I also have to say there is a lot of work made by great people in the lab. As I say, Amanda, for instance, is a co-author of this article. There are other members there. But it's quite a I don't know. I think it's quite a pretty work. But yeah, for sure, for next time we'll have other things to talk about.
Okay then. Thank you and see you.
Thank you. Bye.
Bye. Also, I want to thank you for all of you for your interest and for your time, because you were waiting until now for our conference. And thank you for joining us today. So we are at the end of today's session, and tomorrow we will continue at 2 p.m. In Turkish time, of course. And tomorrow's sessions will be in Turkish because our lectures are Turkish. Yarınki sunumlarımız Türkçe olacak arkadaşlar. Bugün bize katıldığınız için çok teşekkür ederiz. Zamanınızı bize ayırdınız. Bizim konferanslarımızda bize eşlik ettiniz. Ben bu arada arkadaşlarımdan şu an arka planda olanları buraya davet etmek istiyorum. Bugünkü ekibimizin tamamı burada değil ama eksiklerle de olsa hepinize bugünlük veda edelim. Yarın görüşmek üzere diyelim.
Çok
Eklemek
teşekkür ederim.
istedikleriniz var mı?
Konferansımıza katıldığınız için. Gerçekten çok dolu dolu geçirdik bu senede. Yarın saat 2'de Hasan Hüseyin Esenoğlu hocamızla beraber konferansımıza devam edeceğiz arkadaşlar.
Görüşmek üzere.
Görüşmek üzere.
Görüşmek üzere.